Let me just. Oh yes, okay. See, Melina has the marker. Oh yeah, get like Melina, get a paper and a marker. Jess, can you text the group chat that today's our last class? I don't have my phone right now.
Okay, yeah.
Do we need just a normal piece of paper?
What kind of marker?
Yeah, just a white piece of paper.
What kind of marker?
Oh, it doesn't matter.
Mm-hmm.
You can, as long as, yeah, just any marker. And yeah, okay. You can draw, how about, okay, I'll wait, I'll tell everyone's here and I'll tell you guys what to do. Oh, you'll also need the cell cycle worksheet.
Oh, my camera's like.
Oh, your camera's yellow.
I know. Let me fix it. Sorry. Yeah.
Okay, did you text the group chat?
Yes,
like, everyone?
I did. Yeah.
Where is that piece of paper?
Okay.
Because I can't find it. Where is that piece of paper that you want to?
Yellow folder.
Yeah, I have my folder.
Yeah, with that folder, there should be only one more. There should just be the cell cycle. It should, a worksheet. It looks like there's like 6 boxes with like circles inside.
Found it.
And it says stage and whatever. Yeah. I'll put in the chat for everyone to get that. Hold on.
I have that.
Untitled. Okay. Everyone get. Actually, you don't have to. Okay, we'll probably wait like 30 more seconds. I'll just cut.
Is yellow an okay color for marker?
Yellow, sure, if you want. But it might be a little hard for you to see, as long as you can see it. Yeah. Okay. Jess, do you want to share a screen or do you want me to share a screen? I mean, sure, whatever.
Wait, do you get my message? Wait, why is my camera not working again? Sorry.
Yeah, Jess is.
Yeah.
Okay, yeah, you can try to share a screen.
Okay.
While we wait, did anything fun happen this week? To anyone?
My friend's birthday party was yesterday.
My snake laid an egg.
Oh, nice.
Oh my gosh. 2 very exciting things.
Oh my.
Your snake laid an egg. Wow.
Yep. I don't know if I'm going to keep it or give it to a friend yet, but I think it's big enough that I could keep it. Like this, the enclosure is big enough. I'm just going to keep it probably.
Melina rode her bike. Very cool. don't you have to like keep the egg like? Are you gonna like hatch it like, or
You
how
have-
does it even work? I don't even know how it works.
Okay, I'll explain. So you get you get a heat lamp. Of course, you have to have a heat lamp, and you need like some things that would insulate the heat around the egg. It needs to stay warm. So the egg itself, like around the egg, it needs to stay over 80 degrees and above.
Oh,
Between
nice.
80 and 100 degrees. So you have to have a heat lamp and like blankets and stuff. And if it gets too hot, you need to get a fan and whatnot just to make sure it stays alive. So yeah.
Oh wow.
That's exciting though. Okay. Okay, Jess.
Okay.
Yeah.
So today we'll be learning about the inheritance and cell cycle. So first, a bit about chromosomes. Has anyone heard this word before? Anyone?
So you get chromosomes are basically like DNA and whatnot. You have, I think it's 23 chromosomes from your mom, 23 from your dad, and that's basically what you are. It's just a clump of cells,
Mhm.
chromosomes and whatnot.
That is fairly accurate.
Nice.
Kind of, yeah.
So, there's three things that you have to remember. These three like big things. DNA, that is what contains the genetic information. I'm pretty sure you already know what that is because we've been through it. Chromosomes, they're basically DNA that is condensed into vertical clumps. So it's basically like the same thing, but it's like clumped together. So when DNA is like put together, that those like twisted helixes, those are called the chromosomes. And then genetic information, that's chromosomes that determine what our appearance look like. So our genetic information is all got to do with the DNA, and that can like determine a lot of things about. That's what makes each and every one of us different. So yeah, do you have anything to add to that?
Nope.
Okay. So, karyotype. This is like you said you were. I don't know who said this, but you said, because I can't see you, but someone said that
Brianna.
each person has like 23 pairs of chromosomes. So we display it on a chart like this, and this chart is basically called the karyotype. And so different animals have like different numbers of chromosomes. Humans, in this instance, we all have 23 pairs. So these are like this picture, that's the human karyotype. And then, but if you look at like different animals, they have like different amounts of chromosomes. I'm pretty sure like some animals have less than us, some have more. I'm pretty sure.
I think they only have 17 sets of chromosomes, as far as I know.
Yeah. Some of them have like 200, I think. But anyway, it's not because, okay, think of it this way. It's not like if you have more pairs of chromosomes, you have more genetic information. That's not true. It's just that has nothing to do with it. So yes, and then you look here at this. I'm not sure if you can see my mouse, but the XY chromosome at the bottom right, that's those two, they determine if you're a boy or a girl. So yeah, if it's X and Y, then you're a girl. If it's two Ys, then you're a boy. So yeah.
Wait, no. Go back to that. Go back.
Wait,
that one.
did I say that wrong?
XY is a boy. 2 X's is a girl.
Oh, okay. Sorry, I said that wrong.
Yeah.
Okay.
So we all have 2 X chromosomes, but this is a karyotype of a boy because you can see the X and Y.
Yeah.
Interesting. Okay.
Genetics. This is the guy that did the pea plant experiment. So basically what he did was he took the stamens from the purple flower, and then he like did this, he like bred like these purple flowers and this white flower. And then there was like these pods, and then from the pods he got like, you can see, this is the experiment. So he bred purple flower, white flower, and then the next generation only had purple flowers because purple was the dominant gene, so all of those had purple. And then when he bred it again, he found that some 705 are purple and 224 are white. So like because these are like heterozygous purple plants, so it's like big. I don't think we've like talked about that. So it's like
This is too complicated.
okay.
But all you need to know is that sometimes certain genes like mask others. So if you look at like, for example, like your hair color, maybe, does anyone's parents have two different hair colors? Or is everyone's like mom and dad like the same hair color?
So my dad has brown. No, my dad has black hair color.
Yeah.
My mom has like dirty blonde hair, but for some reason I have black hair, and then my sister has light brown hair. So, yeah.
Okay, that is because I'm pretty sure the black hair color gene is more dominant, so it's
It's gonna show up more.
Yeah, it's gonna.
But I'm pretty sure hair color is determined by like multiple things. It's not only like one thing. It gets really complicated. But we're learning about simple genetics here. But just like for hair color, I guess for us, it's like certain genes will show up more than others. Yeah.
Yeah. So, Mendel's pea plants and genetics. So I'm not sure if the sound will work if I play this.
Okay, just try it.
Can you hear it?
Yeah.
Oh, really? Nice.
Okay, go full screen. I can hear it. Yeah.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited. But it all started with Mendel and his peas.
Okay, so that video might have been confusing. If it was, then don't worry about it, but it's just interesting for you to like watch to see what happens.
It reminded me of snakes.
Yeah. I think I heard this concept before discussed on a film theory video.
Really?
Yeah.
That's cool.
It was the video. I think it was the video was adopted because of he looks nothing like his parents, because his parents are, one of them is round and brown and the other one is square and brown also.
Mhm.
So then he ended up going back for SpongeBob's genetics from the episodes of SpongeBob, finding out that he had a square grandfather or great grandfather or something. And then he did this genetic breakdown with the chart that was shown in the video with
Yeah.
the probability of him coming out like that. So yeah.
Okay.
I just want to know what we're doing with the gummy worms.
We're yeah, we're getting there. We just wanted to show you guys inheritance because the gummy worms is related to how you inherit things. It's kind of interesting. Does anyone know their blood type? No one knows their blood type. I mean, maybe you should know it because
I think I'm A.
You're A. Do you know your parents' blood type?
I have no
I
idea.
think I'm trying to remember what it is. Wait, I have it.
Because blood type is also, it's inherited very interestingly. You can, if you know your parents' blood type, you can know yours.
I'm O negative.
You're O? O is rare. You've heard that, right? O is pretty rare.
Yes, because I
Just
think I'm what's called the universal donor. I am O negative, so
yeah,
basically anyone that's dying can take my blood and it will work fine.
Yes.
yeah, that's true.
My mom's O negative and I'm O.
That's cool. Yeah, O is like pretty rare because if you the video briefly mentioned recessive and dominant alleles, but O is recessive. So in order to get it, you need to have a recessive somewhere long. You need to have a recessive parent, which is rare than a dominant one.
Both of my parents are O negative. Oh no, my mom's O negative. My dad's just O, so I'm O negative somehow. I don't really know.
Yeah.
Yeah, that's pretty cool. Most people are A or B or something. I don't even know my blood type. You guys should all know your blood type.
There's A positive, A negative, B positive, B negative, O positive, and O negative. I think
there's AB. Everyone should know your blood type just in case you get an emergency or something. People because if you have if you're A and then
it's like
what?
But you don't know your blood type.
Yeah, I don't even know mine. I'm getting
your blood type.
But yeah.
I donated. No, not I donated. I was to get my blood work. So, yeah. But if you know your blood type, then basically, if you're dying or something, they know what blood to give you then. So that's a plus.
Yeah. Anyway, now we're going to talk about homologous chromosomes. Jess, you want to?
Okay, so the homologous chromosomes, they're basically two pieces of DNA within a diploid organism which carry the same genes, one from each parental source. So you can see that in this diagram. These are basically homologous chromosomes. It's the red ones, they're from the dad, and the blue ones, they're from the mom. And then, yeah, basically, half of it is from the father and half of it is from the mother. It splits it up, so you get both of their genes.
Yeah, this is in your bodies right now.
Yeah.
Half of your eye color genes are from your mom, half of your eye color genes are from your dad. Okay, so it's
Mm-hmm.
Okay, I'll do this one. So basically, you have a bunch of different cells in your body, right? You have nerve cells, you have your blood cells, you have your bone marrow cells, you have so many different kinds for different parts of your body, right? So how did these cells come about? They all started from like the same cells. Like when your mom was pregnant with you, right? At the very beginning, all your cells are the same, and then they specialized into different kinds of cells in your body. And how do they specialize? Like, how do the cells like replicate themselves? That's through the cell cycle. And this is a cell cycle. And this M phase is basically where the cells actually replicate. And G1, S, and G2 are the phases where the cell prepares for replication and division. And M phase actually divide. And then once they divide, they can specialize. So for example, this one specialized into a nerve cell. And then sometimes, like for example, like your liver cells, they go into G0 phase, which is basically where they don't divide unless stimulated. Like for example, I think it's like if you drink too much alcohol or something, I can't remember, then your liver cells are stimulated to come out of G0 phase to replicate, because like alcohol destroys your liver. I can't remember what it was. But something like that. Okay.
So mitosis. So the first step, you have to create identical daughter cells, and you have to also grow. And so the two things that mitosis does is it can create identical daughter cells and also grow and repair, like because cells, they have a life. When they die, you need to replace the cells. So yeah, that's what mitosis is used for.
Yeah, like when you get a cut and your blood comes out, then you need your blood cells need to like replicate themselves, to replace the lost ones.
Mhm.
And we're talking about how the cells divide. Okay.
Yeah. So mitosis, there are different phases of mitosis. Interphase, this is the very start where DNA is duplicated before the mitosis. So it's basically, you can see the chromosomes inside. And then prophase, this is homologous chromosomes form. And then the nuclear envelope also dissolves. So you can see they like duplicate themselves and it becomes double.
And the red one is from a parent and the blue one is from another parent, just saying. Yeah.
Yes. And then metaphase, it's homologous chromosomes line up in the middle of the cell. So like the cell sort of like expands and they go to the middle. And then centrioles connect to the centromeres of the chromosomes. So basically it just lines up in like that sort of fashion, and it's prepared to split. And then anaphase, and it's where they're like pulled apart. They're starting to become two different cells to replicate this first cell here. So this is when they go to opposite ends of the cell and they get like pulled apart. And then telophase, this is the nuclear envelope reforms, and then the chromosomes decondense. Cytokinesis, this is a division of the cytoplasm. And then also this phase, the cell splits, and basically we have two cells instead of one cell. So that's what replication is. For
the critical part is like where you had to replicate your DNA here. So this is like where your cells normally, and then during mitosis, you have two copies of your DNA in a cell. So then when they split, each cell has one copy. That's how. Okay. Oh, now it's a video.
Yeah.
You should already know that mitosis is the process of cell division that produces identical copies of cells and is involved in growth, cell repair, and asexual reproduction. When cells divide by mitosis, the number of cells increases, and hence the organism grows. Different organisms have different numbers of chromosomes. To keep it simple, we are just going to look at what happens to one pair of chromosomes during cell division. Before we start, just a quick reminder that a chromosome is made up of two chromatids, and and one from the mother and one from the father. So mitosis, with our special sciencey names, add in. To this day, I just remember IPMAN to remember which order the stages go in. Then just tag cytokinesis into the end. Interphase, the chromosomes duplicate and become two identical chromatids, joined at the centromere. So in humans, it has gone from the original 46 to 92. In prophase, in the nucleus, the chromosomes condense, and in the cytoplasm, spindle fibers form. Metaphase, the nuclear membrane breaks apart. The spindle fibers attach to the chromosomes, and the chromosomes line up at the equator of the cell. Anaphase, the spindle fibers shorten and the centromere divides, so that each chromosome becomes two separate chromatids. Telophase, the nuclear membrane forms around each set of chromosomes. The chromosomes spread back out in their new nucleus, and the spindle fibers break down. In humans, each nucleus has the normal 46 chromosomes again. Cytokinesis. The cell membrane pinches into two separate sets of chromatids into two identical daughter cells with the same number of chromosomes as the parent. So 46 or 23 pairs in humans. So there we have how cells divide by mitosis to form two new identical cells with the special IPMAT, sciencey words included.
Okay. So that was just the video version of how cells divide. And this happens relatively quickly in your bodies, and it's happening like right now in your body. Anyway, now we're going to do a worksheet, and then we're going to use the gummy worms. So can everyone take out their worksheet that looks like this?
I have an interesting thing to say. My sister ate gummy worms, so then yesterday I had to go to the store and buy new gummy worms because my sister ate them.
Oh, what?
Oh, I'm so sorry.
I thought it was funny. No.
Your sister should respect your materials more.
Okay, does everyone have this worksheet?
I don't really mind.
Gummy worms were good though. Yeah. Okay, I'll give everyone like 10 seconds. It was in that manila folder that it should be in there.
Are there going to be gummy worms left after the experiments?
I think so.
Yay.
Okay, so everyone has this. We're going to fill this out. So we're gonna label the stages that we just learned. So for, just as a reminder, this is just another way of looking at it. Interphase, you have individual chromosomes, right? And then in prophase, they duplicate. And then in metaphase, they line up here. Anaphase, they like split in half. And then they get pulled to the opposite ends. And telophase and cytokinesis is when you form the two opposite cells. I mean, two different cells, identical cells. So I guess we can just, for this first stage, you see a bunch of chromatid, or you just see a bunch of like scraggly lines in the middle. I mean, yeah, scraggly lines in the nucleus of the cell. Which phase do you think this is? The one where you don't really see any chromosomes forming. Yeah, it's one of these. It's either interphase, prophase, metaphase, anaphase, telophase, or cytokinesis. I think it should be. Okay, I'll just tell you guys, it's interphase. The first one is interphase because nothing is happening yet, okay? So if you look at the top left box, just label this stage as interphase. And if you don't know how to spell it, it's up here. interphase. And then the next one, if you move to the right, you see like the X's, right? And these X's are duplicated chromosomes. And in which stage do you see the X's like paired up? Just like next to each other.
Prophase?
Yeah, correct. Okay, good job. So that one would be prophase, the one with the X's, the second box on the top row. And then the third one, you see like the X's again, except they're lined up vertically, and you see like spindle fibers attached to them. And this one, they're preparing to split. So in which one do you see it?
Metaphase?
Yes, good job.
That one looks like metaphase, yeah.
Yeah, it's metaphase.
I was about to say that.
Okay. And then the next one, if you go to the bottom left picture, they actually do split. The X's split up, right? Which phase is that one?
Anaphase?
Correct. Good job. The sister chromatids are pulled apart. Oh, you guys don't know what sister chromatids are.
Well,
They're
you
just
can
half of
actually let people see what's going on and let other people answer.
Okay. You guys can just shout it out. It's fine. Let's see. Okay. I'll share this tab then. Maybe this will be easier because I can't. Okay. Why is it? Now for this stage. The one where it looks like they're in separate cells, but not really. The cells haven't split completely yet. You still see like it's starting to break in half, but it's not really. So, well, we've eliminated all the rest except for cytokinesis, except for telophase and cytokinesis. So, Willa, how will you answer this one? Is it telophase or cytokinesis? Which one do you think it is? The second to last one.
I think it's telophase because in that picture, they haven't split apart yet.
Yeah, you're right. Okay. And then the last one, process of elimination, cytokinesis. You have two identical daughter cells. Okay. And then, yeah, did someone say something?
I have a really strange question. Maybe it's not strange. How do twins work then? Are they just like the exact, like identical twins? Are they just like the exact same setup of chromosomes and whatnot? Just
Identical twins? Yeah, I mean, no one is exactly alike because like, you know, identical twins, they don't have the same personality, right?
like as it goes with like genetics and whatnot.
Well, okay, the thing is different chromosomes code for different things. So for example, like maybe chromosome 22 codes for like how impulsive you are, for example, I don't know. Yeah. And then chromosome 19 codes for like a hair color. Chromosome 19 might be the same for both of them, right? But chromosome 22 might be different. Like, okay. Yeah, but identical twins, I would think probably have, you know, more similar chromosome setups than, you know, other people.
Yeah.
But that's a cool question. You know identical twins?
My mom's an identical twin.
Oh, that's really cool. You guys should get like their karyotypes and compare them. That'd be really fun. That'd be pretty fun.
Yeah.
Yeah, I don't know where you could do that, but that'd be cool.
My mom, like, I don't know, because I've been thinking about it, because I just asked my mom, she's an O negative, just to be sure. She is. Neither of her parents are O negative, and I remember that I was with her sister a few weeks ago when she got blood donated, and she's like an A positive or something. So it's just so random that my mom's an O negative, and I'm convinced that she's adopted.
Yeah, maybe, I think your grandparents probably had the O allele as it was hidden. So your grandparents are probably carriers of the O allele. And then just like randomly, your mom just got both of the O alleles, and then
That
which is which is like a 1 out of 4. It's like not a very high chance, but she just got it. Okay. Yeah, you're interested in heredity and genetics. You know, there's like different jobs, like there's genetic counselors and stuff like that you can do when you grow up. Okay. Now we're going to take out the gummy worm.
Yes.
So does everyone have a bag of gummy worms?
I have 2 bags of gummy worms, but I just ate one of them.
I have a bag of gummy worms.
Okay, cool.
Are we literally just going to be lining up the gummy worms?
Everyone go wash your hands probably first before you touch the gummy worms. I don't know. In case you want to eat them later, I would recommend you wash your hands first. I'll give you guys like 2 minutes to do that.
I washed my hands.
Okay. The gummy worms will basically represent your chromosomes, and we're just going to model the cell
I
cycle.
understand why you use gummy worms instead of something else. Because if we're not actually going to do damage to the gummy worms that will prevent us from eating them after we're done with the experiment, then that means we can eat them.
Yeah. You have a lot of gummy worms to eat. I wouldn't eat all of them at once. You might get sick. Maybe
Why
eat,
would I eat them all at once?
I know.
I know.
Yummy.
Okay. I gave everyone a minute. Okay. We're just gonna watch this video. Basically, he's gonna show us what we're doing.
So during the interphase, your nucleus will look just a solid color, so something like this, and you won't be able to distinguish or see any chromosomes in there. During prophase, you actually start to see the chromosomes condensing, and so you'll start to see these light and dark spaces. The dark spaces are the chromosomes, the light where there are no chromosomes are where they've condensed. So this is prophase. And also what happens is you get these you get the spindle apparatus is forming, and what these are going to do are connect to the chromosomes. So this is prophase. During prometaphase, what happens is the nuclear envelope dissolves, and these start attaching to the chromosomes. So some would attach right here, just to one side. And on this side, they would attach to the other side. So this is prometaphase. Okay? Prometaphase, you have attachment, the nuclear envelope dissolves, and attachment to the chromosomes by the spindle apparatus. During metaphase, what happens is they line up in the middle. They meet in the middle along this imaginary plane called the metaphase plate. So this is metaphase. During anaphase, the spindle apparatus starts to pull, as well as the cell starts to elongate, and these then separate. The chromosomes separate, and they are pulled apart. Okay, so this is anaphase. During telophase, okay, the spindle apparatus isn't needed anymore. During telophase, they form 2 new nuclei. Okay, and now the cell itself is starting to prepare to separate. These are nuclear envelopes. The chromosomes are surrounded by them. And then it's preparing for a process called cytokinesis, which is the splitting into two cells. This is telophase. So to bring it back to our original question about hair, hair is not alive because the parts on the end are not receiving a blood supply and are not going through any cellular functions. The reason why hair continues to grow is because at the base you have cells that are continually going through mitosis, and they're adding a new layer of cells every time. A similar process happens with your fingernails and your skin and other areas of the body. All cells go through mitosis, but these cells go through mitosis more than others. So lastly, I want you to quiz yourself. I'm going to put up a picture, and I want you to make the model yourself, or just tell me what the phase is. So what is this phase?
Okay, we're not going to quiz ourselves. I don't know why. Okay, we're going to. So can everyone take out a piece of paper and a marker, and you're just going to draw.
We already had the paper and the marker.
Yeah, you haven't taken it out yet. Draw 6 circles, 6 really relatively big circles on the paper. So like 3 big circles at the top, and then maybe 3 other big circles on the second row to fill up the entire paper. So like kind of like what this person did. You just have 6. Well, they had. Well, yeah, you just do that. 6 sections. Split up your paper into 6 sections. You're going to represent the 6 different phases. It doesn't have to be circles. You can do boxes. I don't know. Up to you. Okay. Are people still splitting their paper into 6 different things, sections? Okay, people aren't. Okay. Now we're going to do the first phase, which is prophase. I mean, not prophase, interphase. An interphase. Does anyone want to tell me what happens in interphase? The first phase?
Doesn't nothing at all happen in the first phase because nothing can form?
Basically, yeah. Basically, not much happens in interphase except for like DNA replication. So for that phase, for the first circle, you can just put a bunch of gummy bears there. Gummy bears.
We don't have any gummy bears.
Sorry, not gummy bears. Gummy worms. Gummy worms. My bad. You can put like maybe 8 gummy worms. Like this person has 8. Just put them there, and then yeah. And then we have the next phase, which is prophase. What happens in prophase?
They start to like form X's.
Yes. So that was right. So now you guys can just like, you don't have to do an X. You can just have them like, you can either have them crossed over each other, or you can just like line up 2 together, like have some pairs. Or you can actually like do a physical X where you put them over each other. And then during this phase, the spindles kind of start forming, but you don't have to draw that yet. And then after you guys are done with like showing mitosis, I want you guys to take a picture and send it. Anyway, after prophase, after the X's, you have metaphase. And during metaphase, what happens? You guys can put that.
They start to like go apart from each other.
In metaphase, they start to line up, yeah. They don't go apart from each other until anaphase, but during metaphase, they just line up. Like if you look here, they're just like lining up in the center. And then you can also use a marker, use your marker to like draw the spindle fibers, just like connecting to each X, like that. Because these spindle fibers are holding the chromosomes in place inside the cell. They're holding them like this in this vertical way. Make sure you're using like new gummy worms for each section. Don't be like moving the
Well, now I have
gummy worms.
enough gummy worms of the same color for that.
They don't need to be the same color. Well, I mean, if you want to, you can have them the same color, but they don't have to be. We just want to see like how the chromosomes are moving. Okay. Is anyone still working on creating their metaphase, which is this one, the third one? Okay, we're going to move on to anaphase. Who wants to say what happens during anaphase?
Your cells break up.
The chromosomes break up, yeah.
Your
Not the
cells
cell yet.
your chromosomes are breaking.
Yeah.
It
Yeah,
shows
the
the
chromosomes
entire
are breaking in half.
process.
If your cells break, there's a problem. Go to the doctor.
During anaphase, they're just like, it's like they're like this. They're just like breaking to opposite sides, and the spindle fibers are like
Every single gummy worm I have on the first circle and I'm piling every single gummy worm that I'm not using for the other stages on the first circle.
Oh my gosh, it's gonna get crowded. So if you look here, there's no more, the spindle fibers are like pulling them apart. So for this fourth circle, you just want to draw like them. It seems like they're pulling them to the opposite ends of the cell, the spindle fibers. Oh, and we haven't talked about this green stuff. This green stuff is basically where the spindle fibers are made, where they originate from. They kind of like just like come out from these green things, which are called centrioles. Okay, anyone still working on anaphase, on them being pulled apart? If not, we're going to go to telophase. Who wants to tell me what happens in telophase? Okay, it's basically where you have all the chromosomes are in their own like little nucleus thingy, but the cell hasn't finished splitting in half yet. The cell still is like kind of connected here, you see, but it's starting to split in half. But the chromosomes are on opposite ends now. And you might want to label each phase on your paper too, just in case you get confused. You guys will be learning about this in a couple years. I can't remember when, 8th grade, I think. So you'll know a little bit about how it works. After telophase, we have cytokinesis. What is cytokinesis?
They start to form groups.
Oh yeah, you can say groups, or you can just say that the cell just splits completely into separate cells, into 2 identical cells. So you can see like they each have their own nucleus, and you have 2 circles, 2 complete circles, and there's no more spindle fibers. After you guys are done creating these and labeling each phase, can you just if you have a phone, or ask your parent to take a picture, and then take a picture of your completed masterpiece and send it to either Jess or I, or the group chat. Jess, you want to also text the group chat to do that?
Yes.
But this is basically how your cells divide. This is how your hair grows when you I don't know, when you cut your hair, right? You know, when you go to the barber and you cut your hair, but then it grows back really long. It's because of your cells dividing, your hair cells dividing. And then millions divisions later, you have long hair again. If you in high school, you get to work with microscopes and stuff like that. You might even be able to do that now, and you can see the process of cell division under a microscope, which is really cool, I think. But yeah, you're done.
Yes.
That is all we have. Yeah?
Yeah, so this is basically the end of our program. We hope you learned something.
Yeah. And learn something, yes. But thank you for coming. We hope you enjoyed it and had some fun and learned some stuff and got more interested in science, which is very exciting.
Yes.
Okay.
Okay.
Bye.
Bye-bye. Do we end the meeting?
My mom took a picture of my gummy worm pile thing.
Okay, thank you. Can
Nice,
she send
nice.
it to one of us?
Can I eat them now? Or 2 of them?
Yeah, if your parents let you, then sure.
Yes.
Does anyone have any questions or anything? Or want to talk about anything? Science or whatever. You can stay. But if not, you guys are free to go, and thank you for coming.
Yes, thank you so much.
Yeah.
Yep.
And okay, maybe we should just end it.
Yep. Thank you. And this is then. Bye bye.
Okay, bye.
Bye.